Very quickly, let's look at the summary slide real quick. And essentially, what you should remember for this part of the lecture is that this is the electron transport chain, and it's made out of complex 1, 2, 3, 4. And complex 5 is this ATP synthase protein. Usually we call it ATP synthase, so a lot of times you will not see the name complex 5, but you know, it's sort of part of the same system to generate ATP from NADH and FADH2. Alright, so what you should see is that in both complex 3 and 4, you will have proton pumping, okay? And in complex 1, you also have proton pumping, whereas in complex 2, you don't have proton pumping. So this is the reason why NADH can give you 2.5 ATP per 2 electrons, whereas FADH2 can only give you 1.5 ATP per 2 electrons. Okay, makes sense? Because complex 2 does not pump protons, okay? So complex 2 does not pump protons into the intermembrane space, okay? So as a result, FADH2 only gives you 6 protons into the intermembrane space, so then you only get 1.5 ATP per FADH2, okay? And whereas NADH, because you have a conformational change for complex 1 enzyme, as the 2 electrons pass from NADH to Q, right, to coenzyme Q, and because the conformation change, it will promote protons from going from the matrix side to the intermembrane space side, okay? So as a result, you will generate this proton gradient, okay? So for NADH, you can get 10 protons into the intermembrane space. For FADH2, you only get 6 protons into the intermembrane space. Okay, so this is the reason why you get 2.5 ATP and 1.5 ATP respectively. Okay, make sense? All right, so we started talking about this whole idea of proton motive force. Again, this is not a force exactly. It's more of a potential energy across the membrane. Right? Because the proton pumping from complex 1, 3, and 4, a proton gradient, okay, which is the same thing as an electrochemical gradient, is formed. Okay? And this separation of charge, okay, difference in pH, provides the driving force necessary for ATP synthesis. Okay? Right? Because you have a potential energy, and we can then turn that potential energy into a chemical energy stored in ATP. Make sense? Okay? So this pH difference between the matrix and the intermembrane space, it's about 0.75. And for every proton transferred back to the matrix, you can calculate that, and it's about like minus 21 kilojoules per mole. Okay, so it was observed. So then if you have 10 protons stored in the intermembrane space, that's about 210 kilojoules per mole of energy stored. Okay. However, what you should notice is that 210 kilojoules per mole, right? And every mole of ATP is 30 kilojoules. So in theory, you should have gotten something like 7 moles of ATP from the 10 protons. However, what we only see is that you only get 2.5 ATP from NADH. Okay, so you should think about why, right? Because that's something that you should develop the sense for. Okay? All right, so really I want to finish discussing our ATP synthase. Okay, so it's ATP synthase. Typically, the ATP synthase that we know nowadays, there are several kinds, but primarily we're looking at this F1, F0, or F1, F0 type. All right? And ATP synthase is composed of many subunits, okay, but it's going to form into 2 portions, or we usually call it 2 portions, where you have a portion called the F1 knob, okay, so like the doorknob that I mentioned last time. So it's really physically a doorknob that can rotate. And it's connected to the F0, okay, F0 base. Okay, so this base part does not move. Okay, so this part does not move. So if I wasn't clear last time, This part essentially does not move. Okay, what moves is the gamma subunit. Okay, so the globular F1 knob contains 3 alpha beta dimers. Okay, that's not super important for you guys right now, but you just have to know that there are 3 functional sites for ATP synthesis. Okay, and they are arranged about a central stalk, which is composed of subunits G, D, and E. Okay. So the F naught base is composed of 10 to 12 C subunits, okay? And you saw it last time that C subunit is the part where protons can move, right? And 1 subunit A, okay? So it's in on the side, okay? So it helps to anchor your C subunits, okay? And the peripheral stalk, okay, is attached to the F naught base via subunit at the subunit A, okay? A. And at least 4 other minor membrane embedded subunits that are not shown. Okay, so this is just to show you that ATP synthesis or ATP synthase is a really large protein. Okay, and what's really important is just that the central stalk and the C ring composes the rotor, okay, of ATP synthase, and the remainder of the subunits make up the stator, okay, a structure that prevents the rotation of the 3 dimers of F1, so that the 3 dimers of F1, okay, so the alpha beta dimers, they do not rotate. Okay, so if you see the subsequent pictures, they are stationary. What rotates is the gamma subunit right here. It's this thing. Okay. All right. So this is the model for ATP synthesis. Okay. So again, this is your C subunits. Okay. And there's a channel right here for protons to go through it. Okay, and protons want to go through your ATP synthase because remember you have a separation of concentration. Okay, so you always want to go from highly concentrated side to a lower concentrated side. Okay, so using this potential gap, and you essentially give it a route, so your protons will start to move in this direction. So every proton come in, it's going to push the next proton, push the next proton, push the next proton. Okay, so you're going to essentially cause the movement of protons, and as the protons are moving in this channel of C subunit, it will cause a torque sort of on the gamma subunit. Okay, so you're going to turn the gamma subunit on top. Okay, because you're pushing it, so essentially your proteins are being pushed and keep on being pushed. So it's going to sort of rotate your gamma and epsilon subunits on top. So ATP synthase. Remember I mentioned last time that ATP synthase has 3 conformations. Okay, there's the loose, tight, and open. Okay, so you have 3 conformations. All right. So the way it works is very simple. These 3 conformations will change as your gamma subunit turns. Okay, so your gamma subunit is turning. It's going to sort of change the conformation of your alpha beta dimer. Okay, so you have 3 alpha beta dimers, so that means that you have 3 active sites for your ATP synthesis. Right? So for example, if we look at the top third of the enzyme real quick, okay, so this guy is at the open state. Okay, it's at the open state. So when it's at the open state, your ATP can come out, your ADP can come in. So it's sort of freely diffused for coming in and coming out, coming in and coming out. Okay? So of course, you will get ATP going in. Okay? ADP, it's going to sort of diffuse into the enzyme. Okay? And then as your gamma subunit is turning, it's going to turn this portion of the enzyme into the loose configuration. Okay? So this guy is the loose configuration. And in the loose configuration, essentially what happens is you will bind ADP. Okay, so essentially you'll capture the ADP. Okay, and then when you get to the tight conformation, okay, so it's going to do its business, rotate and stuff. And when you get to the tight conformation, it will grab, okay, it will grab ATP tighter than it grabs ADP. Okay, so it says it right here. Okay, the enzyme, ATP is bound on the enzyme. Okay, because the enzyme's affinity for ATP is about 10 to the 7 times stronger than ADP. Okay, so therefore, your ATP is stabilized significantly, whereas ADP is less. Okay, so this will cause you to reduce the delta G of formation for ATP from plus 30 to about 0. Okay, so you're going to change ADP to become ATP. Okay, so this is because when in the tight configuration or conformation, okay, in the tight state, the enzyme is going to promote the change of ADP to ATP. Okay, so after tight, and it's going to become open again, and then ATP is released. Okay, so it goes from open, loose, tight, and then open, loose, tight. All right. So here's the in vitro experiment that shows the rotation of the gamma subunit. Okay, so it turns out what people were doing was instead of purifying the entire ATP synthase, which is difficult because it's a protein, a membrane-bound protein. Okay, so as you guys know, any membrane-bound protein, because it's membrane-bound, right? So it's going to be soluble in lipid state. Okay. But in this case, we don't want that. We just want to see the effect of rotation, so you don't need the C ring. Makes sense. So here we only got alpha beta dimers and the gamma subunit. Okay, and on the gamma subunit, I can attach a actin filament, and this is done through strep-avidin association. Slight detail. But essentially, you have an actin filament, and we can label the actin filament. Okay, and here. There's no proton gradient, obviously, because there's no separation, right? There's no membrane, so no proton gradient. So here, what we're going to do is we're going to feed ATP, okay? And essentially, what we want to see is your ATP synthase reversible, okay? Can we use ATP to generate a rotation, which then, you know, we can sort of say that maybe it can create your proton gradient. Okay, so here we just want to see that whether or not you can use ATP as a substrate instead of a product, okay, and rotate ATP synthase. Okay, so the reversed reaction of ATP synthase, okay, reverse direction. We want to use ATP, and the results show that yes, we can. All right, so you can see the little thingy rotate. Okay, it rotates pretty fast. The time between each image, it says that here is like 133 milliseconds. Right? So not too bad. Okay, not too bad. You can actually show that your ATP synthase is reversible. Okay. Yeah. So when you have time, you can go check these videos. All right. So the reversibility of your F1, F0 ATP synthase. Right. So if you look at the two pictures on the top, you will see that. In the first picture in A, okay, this is the normal respiration chain. Right? This is the normal electron transfer chain, where you generate your proton gradient, okay, because you have O2 here. Okay, so when you have molecular oxygen serves as the terminal electron acceptor, which then will facilitate your entire electron transport chain. Okay, and as the proton coming back down. Okay, you generate ATP. And the question is, right, how does your ATP come out, right? Because we said that you do this entire process in mitochondria, but then in cytosol, you need to have a bunch of ATP, right? Because how else do you make, I don't know, DNA, or well, DNA is done in nucleus. How else do you make some amino acids or other fatty acids, for example, right? A lot of these cellular processes or moving, okay, your cellular movement, all of these cellular processes require ATP. But if your ATP is stuck in the mitochondria, it's not very useful, okay? So it turns out there is a special transporter called the adenine nucleotide translocase, okay? And this particular translocase will bring in ADP and take out ATP. This is which kind of transporter? We talked about transporter last semester. I'm not talking about like active, passive, okay? There's a special name for this type of transporter. one goes in and one comes out. Uniporter, symporter, antiporter, that kind of stuff. Remember that? Okay, it's antiporter, right? Exactly. So one comes in, the other one comes out. Okay, so this is a way to sort of balance your charges. Okay, right. So you have a transport protein right here. Okay, a membrane protein that sort of serves as a transporter that will take you take out the ATP and bring the ADP. Okay. All right. Now, in the absence of O2, okay, during hypoxia, so for example, when you're exercising intensively, your cells are going to be temporarily short on O2. You do have a reduction of the proton motive force because if you don't have O2, you don't have your proton gradient generation. Okay. So the cells, again, like we said previously, will rely mainly on homolytic fermentation for ATP production. Okay, so it's going to be from the outside. Okay, from the glycolysis. Now, obviously, if ATP is generated on outside, okay, what about ADP? Where do you get ADP coming from? Okay, so this is sort of the reverse reaction of the figure on the left, where ATP can go in, ADP can come out, okay, using the same protein. Make sense? All right. And this process, pumping protons out of matrix, is used to temporarily sustain the proton motive force to support other processes, such as metabolite transport. Okay, so a lot of times, what you have, you still, even though you don't have O2, but you still need to generate a proton gradient. Okay, so here what it shows is that you're using ATP that brings it into the matrix of the mitochondria, and then you're going to do the reverse of ATP synthase, okay, so that you generate and maintain a proton gradient. Does that make sense? Because proton gradient is used for other processes inside the cell. So sometimes if you don't have molecular oxygen, so you don't have a functional electron transport chain, so you don't have build up of proton gradient, but you still need it. So what do? You take ATP from the cytosol, and then you make the proton gradient using ATP. Make sense? Okay, so this is sort of shows the reversibility of ATP synthase. All right. So here I just wanted to show you there are several different mitochondrial transport systems. A lot of these we have seen previously. For example, we have seen pyruvate. Okay, so pyruvate can get transported. And pyruvate transporter is also an antiporter, which takes in pyruvate and it pumps out hydroxide anion. Okay. We have previously also briefly talked about malate, okay, which we'll see in the next slide. Okay. Malate can go into the mitochondria, and this is used for bringing in NADH from the cytosol to the mitochondria, we talked about last time. Okay, so malate, succinate, fumarate, those guys can go in. Okay, and they use this dicarboxylate transport system because they're both, all these three are dicarboxylates. Okay, so we also saw this adenine nucleotide translocase, ATP synthase can pump protons, etc. Okay, so there are quite a bit of different transporters that will transport stuff in and out of the mitochondria. Okay, make sense? So I should have shown you guys this slide before we talked about the NADH shuttle, but there are two types of NADH shuttle. One is the one that we talked about using malate, and the other one is using glyceraldehyde 3-phosphate. Okay, so here the first one, this is the dihydroxyacetone phosphate glyceraldehyde 3, glycerol 3-phosphate. Glycerol 3-phosphate shunt or shuttle, where you can use NADH and you reduce DHAP into this guy is glycerol 3-phosphate. Okay, so again, what you're doing is you turn that guy into glycerol. Okay, so you turn a ketone into an alcohol. Right, so you effectively reduced DHAP. Now glycerol 3-phosphate can penetrate the outer membrane of your mitochondria. And what that guy then can do is donate the electrons onto this flavoprotein, okay, which contains essentially FAD. Okay, and then FAD becomes FADH2, okay, and then FADH2 goes to Q, etc. Okay, so this is one way to do it. And of course, the other way is malate. So last time I mentioned pyruvate, right? Pyruvate can become malate using the malic enzyme, goes into the mitochondria, becomes pyruvate again. Or in mammalian systems, you can use oxaloacetate, okay? But pretty much the same thing. Remember that oxaloacetate and pyruvate is just one carboxylate difference, right? You don't change the redox state. So you can reduce oxaloacetate to malate. This enzyme is called what? Malate dehydrogenase. Okay? Malate dehydrogenase becomes malate. And then malate, you have a transporter. Okay, transporter. And then goes in, becomes malate. And then malate becomes oxaloacetate. This is again malate dehydrogenase, or I'm just in short, right? MDH. Okay, so MDH, okay? So then, malate becomes oxaloacetate. And now this is the thing that I didn't ask you guys to remember at this moment. Oxaloacetate can do a transamination becoming aspartic acid. Okay, so this is a transamination where oxaloacetate this is OAA. What is this? This is aspartic acid, okay, or aspartate, right? So essentially you do a transamination process. You turn oxaloacetate into aspartate, and the aspartate can cross the membrane. But minor detail, okay? So the detail of this is not very important for you guys at this moment, but I just want you to understand that NADH, when it goes from the cytosol into the mitochondria, you have to use a shuttle, okay? Right? So that's the bottom line. Right, and the shuttle, there are two types. You either use the DHAP or you use malate. All right, so this is probably the most important slide, all right? So the most important slide that will sort of summarize everything that we talked about from day one, maybe day two to now, okay? Is that you should be able to answer the question of, all right, if we say that ATP is so important for biological process, 对不对？我们说 ATP 很重要，为什么？因为它可以把一个 reaction 从 positive delta G 变成 negative delta G。OK， 这还蛮厉害的啊，有没有厉害？还好哈，有，还好，可以啦，不错，厉害啦。对 ，delta G 从正的变成负的，那还蛮强的啊，好不好？Right， 所以你可以把 delta G 从正的变成负的，所以 ATP 很厉害。ATP 还可以拿来干嘛？还可以拿来，比如说让你的什么肌肉 movement 啊，什么 motor protein 要动啊。我不知道你们在那个普生的时候，或者是有没有看过一个 video， 一个东西在走来走去，有没有？那要用什么？要用 ATP 啊，不然它怎么会走？对不对？有没有想过这件事？不然它怎么会走？它没事干嘛走来走去？因为它爽，不可能啦，好不好？当然不是，所以因为它会走，就是因为它有 ATP, right? So ATP is very important, right? So ATP is super important for all cellular processes like growth, whatever, okay? All sorts of cellular maintenance, you always need to have ATP, okay? So ATP is sort of the core energy for cells, right? For biology. So essentially through lecture 2 to today, I basically explain how you make ATP from something simple like glucose, okay? And how many ATP can you make from glucose? So if we look at glycolysis on the very top, glycolysis can give you 2 pyruvate, 2 ATP, and 2 NADH, okay? Glycolysis. And we can then turn pyruvate to acetyl-CoA. So you have 2 pyruvate on top, so you can make 2 acetyl-CoA and you make 2 NADH, okay, because 1 pyruvate to 1 acetyl-CoA, 1 NADH. Take everything times 2. TCA cycle, 1 acetyl-CoA can give you 2 CO2, 3 NADH, and 1 FADH2, 1 ATP. So times everything by 2, you'll have 6 NADH, 2 FADH2, and 2 ATP. Add everything up, so the net conversion is glucose to CO2, complete oxidation. And you will generate 10 NADH and 2 FADH2 and 4 ATP. Okay? So the complete oxidation, okay, of glucose give you this much reducing power and 4 ATP. And remember what we just learned in previous lecture and this lecture is that NADH and FADH2, if in the presence of oxygen, you can convert that energy, okay, into the proton gradient and then use the proton gradient to then produce ATP, okay? And for every 1 NADH, you can generate 2.5 ATP. For every 1 FADH2, you can generate 1.5 ATP. Okay? So you multiply those things out, you get about 32 moles of ATP synthesized per mole of glucose. Okay? Right? So you need to have a very clear idea of where are the ATPs coming from. Okay? 所以就很重要嘛，就是说，比如说我会给你一些题目，比如说你今天吃乳酸 ，OK， 一个乳酸可以给你几个 ATP， 你要算得出来 ，OK， make sense? Right, so this is sort of the most important part of Biochemistry II. Like for you to be able to calculate what do you get in terms of energy and what do you get in terms of reducing cofactors for any type of feed. And by feed, I mean you can have glucose, you can have, what else can you have? Glycerol, you can have lactate, you can have acetate or ethanol, right? Could very simply ask you guys a question of, you know, if you guys like to go drinking, for every mole of ethanol, how many moles of ATP can you get? Things like that. Okay, so you should be able to calculate that and understand that, right? Now at the very end here, it says how much energy is 32 ATP worth, okay? And how much energy does glucose oxidation to CO2 provide? How much energy? If we look at a chemical conversion, like you take glucose and you do a combustion, okay, you burn it, how much energy can you get, right? So we talked about that, I think either at the very beginning or last semester, right? They don't balance, right? 懂我意思吗？就是它不会 balance 嘛。然后我们之前不是有讲，一个 glucose 如果我用烧的完整的 oxidation， 把它烧成 CO2， 有没有？它不是会放很多热吗？不是会放那个调查机好像是-2000还是-3000，忘了 ，OK？ 但是你知道说在代谢层面上，一个 glucose 就只能给我32个 ATP， 那32个 ATP 纯的能量就等于-30乘上32，就差不多1000多一点点，所以你中间其实 lost 掉很多 energy，OK？ 那 why？ Right， 就是为了要 provide 一个 driving force， 让你所有的 reaction 的调查机都是 minus 的 ，right？ 因为 otherwise， 如果说你把那个能量完完全全的 balance 住，那你的调查机就等于0啦 ，right？ 那如果你的调查机等于0的话，那你的 cell 就很容易就一直存在 equilibrium。Okay？ 那 remember 哦 ，life is never at equilibrium， 懂我意思吗 ？OK， 我们说过 life 是在 steady state。但是它不会在 equilibrium， 所以那个 biology 是不会有 equilibrium 这件事情，因为如果在 equilibrium 的话，那代表这个生物已经挂了。OK？ Alright. So the rest of the slides are less, I would say, important, but they're good information for you guys to know. Okay? So for example, I don't know if you know this, but the genes encoding the electron transport chain is actually found on the genome of mitochondria. So how many of you guys know that mitochondria has its own genome? Have you heard that before? Right? 有嘛吼? So the electron transport chain proteins are found on the mitochondrial genome. So this is, one of the reasons why people think that perhaps this is during evolution, mitochondria is like a little bacteria that fused with the eukaryotic cell. All right. So because we're talking about respiration and oxidative processes, we need to spend a little bit of time talking about oxygenases, because oxygenases also use O2 as a reactant. Okay. We will get to a lot of oxygenases throughout our class, but for now, this is not terribly important, but you just have to know that oxygenases, you have two types, monooxygenase and dioxygenase. Okay. Dioxygenase just means that it incorporates both of the O2, there are two O's, right? Two oxygen atoms, and dioxygenases will just put those two both into some molecule. Okay? Now, this is a very important process or an important type of enzyme, okay, because they are used to do a lot of things. Like seriously, oxygenases are used to do a lot of things. For example, amino acid biosynthesis, degradation. This is the first reaction in tryptophan degradation, right? Where you want to turn tryptophan to something that you can degrade. You want to open the ring structure, and a lot of times to open the rings, you need to use dioxygenases. Okay. Also, you can have some bacteria that can grow on methane, for example. Okay. Those things are called the monooxygenase, which only incorporate one of the oxygen into your molecule, and the other one goes to water. Okay. For example, there are bacteria that can grow on natural gas. Okay. So for example, things that grow in probably, places where there's a lot of methane. And essentially what they can do, okay, is turning methane into methanol. Not a simple reaction. Okay, why? Methane is super stable. Okay, so anything without functional groups like saturated carbon, those things are typically super stable, right? So then it's not an easy reaction, but yet you have these oxygenases that can do this process. Okay, actually, this process is going to be a very important process in the next 20 years, okay? Because you guys know that petroleum is running out, okay? Petroleum, 你知道 petroleum 石油嘛？油的那个枯竭 ，OK？ 所以现在在很多能源的政策，美国啦，至少他们现在要推的一个东西是用天然气 ，OK？ 天然气，因为什么？美国大陆大概有200年的天然气 reserve， 所以他们未来啊。OK，in order 去得到他们自己的 energy independence， 他们会要去想办法使用 methane， 这是 methane。OK， 那当然一方面为什么不要使用石油？因为石油其实现在我们看到石油 reserve 的量大概剩40年，差不多了。大家估算剩40年。那个之后你的 chemical resource 要从哪里来？所以这是一个很大的问题。那methane 这种东西，就是如果你有200年的 supply， 那你可以去利用它，把它变成其他的东西。因为如果 methane 只是拿来烧，那就有点废，你知道吗？就是你没有其他的 chemical 的 source 啊 ，right？ 那很多人就会需要去想办法，怎么样子使用 methane 把它变成一些其他的 chemicals。那当然有化学的方法。OK， 那其实我们在看做 methane conversion 做的大概最好的，其实就是这个东西。这个东西叫做methane oxidase 还是叫做什么东西我忘了，但是它其实就是一个 monooxygenase。那它就是使用 O2 去把 methane convert 成 methanol。那这个东西可以在常温常压下做。那化学反应的话没办法。这个化学反应的话，通常都温度会比较高，所以很多人利用 biology 的这个特性 ，OK， 然后去学习它的 catalysis 是怎么做的，然后去合成一个那种化学的催化剂，去有点像模仿这个 part， 然后去做到 methanol。因为你一旦做到 methanol 之后，你可以做后续的一些化学反应，然后去做 chain elongation， 就可以做出其他的 chemicals。OK，so monooxygenase 其实是一个蛮 important 的东西。那at this moment， 其实大家对 这个 methane to methanol 的 understanding 还是不够多，所以大家还在研究这个 protein. Okay, another type of protein that is quite important. This thing is called cytochrome P450. Okay, why is it called P450? Because actually, I forgot why it's called P450. I think it has to do with like some absorbance. Sorry, yeah, I forgot. Once a time I know because it's a cytochrome, right? It has a cytochrome inside, so I bet you anything it has to do with, you know, it has a heme or it has a something that absorbs at 450 nanometers. I bet, but you know, I could be wrong. But regardless, okay. Cytochrome P450 is a very powerful type of enzyme. It's not one single enzyme. It's a very powerful type of enzyme. Okay. The reason for that is because they are involved in doing hydroxylation. Okay, hydroxylating a bunch of variety of proteins. Compounds. Okay. Why is that important? Why is that important? Because if you are, you know, aware of like the source of drugs, like if you're interested in developing medicine, okay, 如果你今天是对那种药物比较有兴趣 ，okay， 那你要知道药物是怎么来的。有没有知道药大概是通常是怎么来的？你不要说现在有什么电脑的辅助去设计药物什么什么的，我们先不要讲那个，我们先讲那种可能30年前到1000年前所有的药物是怎么来的。就随便丢嘛，对不对？你就是随便 collect 一堆 sample， 然后就是 collect 一堆有的没有东西，然后就丢给有病的老鼠或者有病的人。再早期一点是人啦 ，OK， 就是有没有？就是这个人重病，然后我也不知道他为什么病，反正我去收集一堆，例如中草药之类的东西，然后就给他吃吃看，你吃吃看，好了，这个可能就有用，有没有？所以那个中国神话有没有那个？对，神农尝百草嘛，就这样来的。所以为什么是百草？就是。植物里面有很多各式各样的 metabolites，OK， 有那种各式各样的哦。那这些各式各样的 metabolites， 很多时候都是靠 cytochrome P450 去做的，去做 modification。怎么做 ？OK， 怎么做？主要是说，当你今天如果合成一个 some kind of 这种小分子啊，这 some kind of 那种分子，那有一些 pathways 可以做出这样子的 ring structure， 但是有的时候你会发现你需要去做，比如说你看这个 molecule， 这个 molecule 到这个 molecule 差在哪里？差在这个 OH， 对不对？那这个 OH 在什么碳上面？它在一个 saturated carbon 身上。那各位就是要对化学有一个概念，就是说 saturated 的 carbon 你很难做反应 ，right？ Make sense？ 因为你怎么去把它身上的那个 CH 棒给拔掉？很难嘛，所以这个时候这种反应，你一般的 enzyme 或是一般的这种 reaction 很难去做，所以这个就为什么 cytochrome P450 type 的 enzyme 很厉害，因为他们可以做的一件事情就是他们可以specifically 在有一些 saturated carbon 身上放 OH， 那你要是可以做到这件事情的话，你就会做到一个不同的 molecule。那这边如果有个 OH， 它可以变什么？可以变 ketone 啊，对不对？可以变成一些其他的东西。Right， 所以怎么样子放 functional group 其实是一个很重要的 process 在biochemistry 里面。OK， 所以这个药物开发早期啦，当然我们说早期的这种药物 discovery， 很多时候都是靠一个 library of 药物。那这 library 从哪里来的？像这种小分子，这么多 ring 的这种 organic molecules， 绝对不是从什么石油来的。OK， 那都是从 biological origins。所有这些东西。Usually 都是某一种 biological origin， 所以就是你都是从生物里面萃出来的。那这些东西呢，当然如果说你有了这个 precursor， 然后你要去合成你的药物你可以再用化学反应去做。但是这个叫 lead drug， 我们说 lead target，OK， But you know, for our purpose, cytochrome P450 is a very powerful enzyme, okay, doing hydroxylation on saturated carbon, okay. Last two slides, I have about like 5 minutes, real quickly. Oxidative stress, okay? Because we're, you know, aerobic organisms, right? So we are breathing in oxygen. Now, sometimes, okay, oxygen obviously is a very oxidated thing, okay? So sometimes when oxygen, right, receive only one electron, for example, you make superoxide, okay? You make an oxygen radical. And obviously, you guys should hear from the news or from your professional knowledge that radicals are not exactly good for you. Yes? 有听过哈？有没有？所以要多吃 antioxidant. 那这个 radical 呢 can become a lot of other things. For example, hydrogen peroxide, hydroxy radical, peroxynitride, oxidized coenzyme Q. Okay, things like that. Okay, so actually, oxidized coenzyme Q, this QH with a dot, if it interacts with oxygen, it can make superoxide. And this happens a lot. Why? Because you have QH radical inside of your electron transport chain. You have oxygen inside of your electron transport chain. Okay, so both these guys are going to be found at around pretty similar locations. So they can react, okay, in a chemical reaction. This is not an enzymatic reaction. Okay, so as a result, you generate a bunch of these things called the reactive oxygen species, ROS, and they are what we generally call oxidative stress. And they're really bad for you because they damage proteins, they damage especially DNA. Okay? Damage proteins and DNA. So there are several enzymes that you can use to combat oxidative stress. Number one is your antioxidant. Okay, this is why people always encourage you to eat things that have vitamin C, vitamin E, those things. Okay? Vitamin C and E. Those are chemical antioxidants. You can also have this thing called the superoxide dismutase. Okay, it's a type of enzyme that will take two O2 radical and you make it into hydrogen peroxide. And the hydrogen peroxide can use another thing called catalase, which converts that into H2O and O2. So these things are sort of together. Or you have hydrogen peroxide. You can also use this other thing called peroxidase, which will use a organic substance, and in this case, it's just a glutathione. It's a type of molecule that is inside the cell, intracellular, that is used as a reducing agent. Okay, so you can convert that, and this is a thiol. Thiol into disulfide. Okay, so this is just some type of stuff that biology has to combat oxidative stress. Okay, so that's it. That summarizes all of the stuff that we have for this third of the course. That summarizes all of the stuff that will be covered on your exam. Okay?